Alright, so this lecture is going to cover the digestion system, and basically you guys probably pretty well know it as how we get that food, how we get those nutrients into the body. We eat all that food so we can get the carbohydrates, we can get the amino acids, the fatty acids, the triglycerols, also ions and so forth. So this is we will look mainly at the macronutrients, the big nutrients like the carbohydrates, the proteins, and the amino acids and the fatty acids. So we'll look at how we break these down and then are successfully able to absorb them. So the two main functions is digestion, hence the name, digestive system, and basically we are doing that digestion so we can absorb. And what we're going to see is most of these nutrients come in longer chain molecules, like proteins are long chains of amino acids, carbohydrates or starches are long chains of carbohydrates, monosaccharides, and lipids are the glycerol with the 3 fatty acids. So to be able to successfully absorb, which is the goal, to get the nutrients from the digestive tract into the bloodstream so the body, the tissues can use these nutrients, we have to first make sure that we break them down. Okay, breaking down to monomers or molecules that are 2 or 3 molecules long. Okay, and we're going to be breaking it down. If you remember from our chemistry days, we did condensation reactions to break them down. We're going to ultimately have to do hydrolysis to break these larger molecules into individual components. When you actually look at the digestive tract, we can actually consider this digestive tract part of the external environment, because those food substances that we eat and bring down through the digestive tract are not into the bloodstream, don't have access to the other tissues until they are actually absorbed out of the digestive tract into the body. Okay. It's important. This is a very harsh environment. We will see some of these enzymes can be very harsh. If they were running around in the body, it could cause some havoc. So we regulate them to this, basically this external environment running through the digestive tract. So what are the overall functions of the digestive tract? Essentially, we're doing digestion and absorption, but to do that, we have to move everything through, and we've got to do several things to get this successful digestion and absorption. One of those things is we have to have motility. We have to be able to move the food substance through the digestive tract. Secretion, because we're going to have many chemicals that are going to be involved in taking that food and breaking it down into those individual or those very small components. We're going to have secretions both of the exocrine and the endocrine gland. Exocrine glands are going to be the substances that are going to help digest that food material, and then the endocrine system is going to be some hormones that are going to help regulate the system as food moves through it. And then finally, we got digestion, which we've already seen. That's the breakdown of food, and we're going to see it's actually broken down into 2 components, into actually mechanically digesting the food and chemically digesting the food. And then once we're there, we can absorb those nutrients. That's the ultimate goal, is to get absorption into the bloodstream so our tissues throughout the body have access to glucose, to all these good stuff in the food material. And we store and eliminate. It doesn't just go in the mouth and out the backside. It is storage and elimination. Once there's enough storage, we go to eliminate. And the other is, it also forms an immune barrier. Just like your skin, it has a layer of epithelial, and it'll have some areas in which the immune system lies, because we are bringing something from the outside environment. Yes, there are good stuff, those good nutrients in there, but there can also be foreign pathogens along the way. So we got some barrier, and we also have some immune cells. We'll talk about some lymphatic tissue lining or close to the lining of the digestive tract that are going to help to attack any pathogen that we may ingest also. Your digestive system can basically be broken down into two major types of organs. We have the organs that make up the actual digestive organs, is those that make up the gastrointestinal tract. Basically, they are the organs that make up that tube that goes from the mouth all the way down to the anus, and that is going to include all these organs and where digestion is going to take place of those food materials and the proper absorption, storage and elimination, so forth. The organs that are not part of the tract but are going to aid the gastrointestinal tract into getting proper digestion and absorption are the accessory organs, accessory digestive organs. We have teeth, tongue, salivary glands. Those are going to physically break down the food. We're going to see the liver and gallbladder are going to be involved in what we call the chemical digestion, in breaking the bonds in those larger nutrient molecules. Okay, so the actual tubing is the GI tract, gastrointestinal, the digestive organs, and then the accessory organs are there to help or aid in the processes taking place in the GI tract. Now let's do our little anatomy. We're going to look at the layers that make up that tube, in which the GI tract. This is set up in a way because of the functioning of this system. Remember, we want to get digestion and we want to get proper absorption. Absorption is going to be, we have to diffuse food into the body, and so we're going to want a large surface area, and that is kind of how the GI tract is going to be set up. But we have 4 tunics or 4 layers to the tract. Okay, there is the mucosa layer. This is the layer that actually lines the lumen, lines the tube. Okay, this is the one that it will be in access to the material moving through it. It will have direct contact through it. It is where we're going to do many of the secretions are going to come from here, because the secretions are going to aid in that digestion and the absorption. And then we also have the layer in which absorption is going to take place. Okay. Just below that is the submucosa. It is a very vascular layer with glands and nerves, because what are we trying to do? We're trying to take the food from the digestive tract and bring it into the bloodstream and be able to circulate it throughout the body. So we need to have a very vascular layer there. Okay, so there's a network of capillaries, all right, and also a network of nervous system throughout there. Okay. Besides that, it is the muscularis. So we're moving out, went here. Okay, the mucosa is the lining, the submucosa is here, and now we're into these muscle layers here. And there are actually 2 layers. Remember, there is the circular that basically wraps around the tube, and then the longitudinal, which runs along with the tube. Okay, and that's going to allow for that motility, that movement of food material. In the stomach, we'll see there is actually a 3rd muscle layer, because it's going to help in churning the food. But you don't have to control these muscles. You don't have to sit there and think, I got to move my digestive tract. So when stuff is not controlled voluntarily, it is the autonomic system that is going to control the movement or the contraction of the muscles through the digestive layer, or the muscularis layer here. Okay, and then the outer layer is the serosa. It's going to face towards the organ or the inside of the body. Okay, and we're going to see it's going to be continuous with what we call the mesenteries. The mesenteries basically are there to hold the digestive organs in place. So here's the mesentery here. This would be attached to the wall of the abdominal cavity, and then we're attached to the GI tract here, and that is going to hold these organs into place. So the GI tract has basically its own nervous system. It has this enteric nervous system, this mesh-like network of neurons. And you can imagine those small intestines alone are about 20, if you wrap them out, would be about 20 feet in length. So you can imagine how much neurons make up this. You can see embedded in the circular muscle here, or in the layer here, the submucosa, right? And these black neurons here. And they almost operate similar to like the autorhythmic cells, because they do have input from the sympathetic and parasympathetic that help modulate the activity of this mesh-like network of neurons. But if these are severed, you'll see that these neurons can actually operate. So they're kind of like the autorhythmic cells that are controlling the autonomic nervous system, control the depolarization. These autonomics are going to modulate the activities of these neurons, but these neurons are also able to operate alone without that neural input. Okay. So you can see we're going to be innervating the muscles here and some of the endocrine or exocrine glands and endocrine glands for secretion. Okay, so this big vast-like network called the enteric is going to help modulate the movement of muscles to help move the materials, and also much of the excretion or secretions throughout the system here. So let's walk our way. You may have done this in your anatomy class, walked our way through the GI tract, but let's walk our way through. First, we'll start at the oral cavity and end up with elimination at the anus, but at the oral cavity. Okay, the first part of the tubing, you have teeth there. Okay, teeth allow for what's called mastication, the grinding down, the physical grinding down of those materials. That is called mechanical digestion. When you're breaking the materials down into smaller physical components, it is mechanical digestion. And that starts with the teeth here and the mastication. Okay, also in the oral cavity, we're going to combine those secretions. Remember from lab, we got amylase up there in the oral cavity being secreted in from the parotid gland. We got mucus, also some antimicrobial agents. So we start breaking down any of the bad stuff that is in the food. Okay. Then we move the food down into the pharynx, and then we are going to swallow. Okay, there are 3 phases in this degglutination. All right, there's an oral phase in the oral cavity, pharynx. So it is a complex system in the swallowing. We have 3 layers. We're going from the oral cavity to the pharynx finally into the esophagus. Once we're in the esophagus, that enteric system is going to move. We're going to move the material with those walls of the GI tract layer. Okay, those longitudinal and those circular layers are going to help move, and we're going to look at that process in a second. Okay, but once glutination, we've moved into the esophagus, and now we're going to start our way down basically that internal part of the digestive tract. So we've now swallowed and we've moved what we call the bolus. Basically, that food-like material is made into a bolus, and we're going to move this down the GI tract. And you're going to see that the GI tract, the muscles, those circular and longitudinal muscles work in a wave-like manner, and basically it starts, muscle contraction will start here and move down here subsequently like in a wave, and so that helps push that bolus through the system. And that movement, that wave-like movement through the GI tract of the muscles pushing it, is called peristalsis. Another form of contraction of the muscles is going to be this segmentation. Segmentation is basically constricting the lumen in different sections. We're going to oscillate between the sections, and that basically brings the material, the food material, back and forth, back and forth, back and forth, and it helps churn up the material. We want to continually break this down. We want to break it down into small components so we can chemically break it down and then get further absorption. Okay, so the segmentation helps in what we call the mechanical digestion, the physical breakdown of the food into smaller components. Okay, so we're going to move down the esophagus, and one of the first big organs we're going to reach here is the stomach. The stomach is going to actually serve several functions. It's going to be a place where we can store food. All right. We're going to initiate chemical digestion. Okay, chemical digestion is going to be the breakdown of these foods. Say there is each of these little balls is a glucose molecule, and they are bound together. Okay, what we saw in our oral cavity is we had our amylase. Our amylase is going to break down starches. Starches are big, long molecules of carbohydrates, of glucose. So we need to first mechanically break down. We were physically breaking down that food material into small substances, and then eventually we have to break down the bonds. All right, and to break down the bonds, we're going to use enzymes. In the mouth, we saw amylase. We're going to start the starch digestion. What we're going to see in the stomach is a new enzyme, pepsin. All right, that pepsin is going to start the digestion, the chemical digestion, the breaking of the bonds, breaking of the peptide bonds between, in this case, in proteins, these would be amino acids. Right. What you've also seen is in the stomach has a high acidity. It has a very low pH. Okay. That low pH is actually going to serve several functions. It's going to help kill bacteria, also unwind some of those proteins, denature them. Okay, because we got to get at the bonds between the amino acids. So we got to get these larger molecules broken down in which we can use the enzymes. The enzymes have to have access to all the molecules. Say the molecule is like this. That's the strand. All right, we have to get into enzymes to break down molecules in there. So we have to get these molecules broken down into small components, so then the chemical or the enzymes can start chemically breaking down the bonds. Okay. What we're also going to see in the stomach is it has 3 layers of muscle. That allows it to basically do churning effect. It's going to constrict in different regions, and it's going to churn the food into almost like a paste now, into a paste called chyme. Okay, so now from this bolus, we're breaking down the bolus or more, and then as we get to the stomach, we churn it up, and we're going to see there are some secretions here from the stomach, and we form this paste-like material with the food. And if you remember from your anatomy days, the stomach has 3 layers I already talked about. We have this third oblique muscle layer that helps in that churning of the food, churning the food back and forth. Also, we have these rugae folds here that allows, when you eat a large meal, allows the expansion of the stomach to be able to store and hold more materials. And what we're also going to see are these folds have these gastric pits, and these pits are going to be lined with cells that are going to have exocrine secretions. Or it's highly acidic. Some of the cells are going to secrete hydrochloric acid into the stomach. Some are going to secrete those pepsin. It's not going to be released as pepsin, but pepsinogen. We'll see that in a sec. Okay, but those gastric pits that line this region here are going to add secretions to the food material coming into the stomach. So looking at some of the cells that line those gastric pits, here is the lining of the stomach here, and the pits lie down in here. There are cells that line these pits. First of all, we'll see the mucus cells. You can imagine, what do they secrete? They secrete mucus, okay, an acidic mucus. Right. This is important because it helps protect the lining of the stomach. Because you can imagine, we have this high acidic, very low pH environment. This can be detrimental to this lining. It can be corrosive. Okay? So this mucus is going to help protect the lining here. Okay? It's a very hostile environment in the stomach with this low pH. Okay? Cells that are actually going to make the low pH or secrete the acids are the parietal cells. Okay? They're going to secrete the hydrochloric acid that is going to make the acidic environment, make that low pH environment in the stomach. Right? And then other cells important in here are the chief cells. They're going to secrete what's called pepsinogen. Okay? Pepsinogen is a precursor to that pepsin enzyme we talked about briefly. Okay. Pepsinogen is called a zymogen. It is released. These cells don't make the active pepsin enzyme, because remember, pepsin is going to break down peptide bonds in proteins. It's going to break down those bonds and those amino acids. Your cells are full of proteins. You don't want an active enzyme in there breaking down, chopping up your proteins in your cell. So the cells make an inactive form called pepsinogen, and then when pepsinogen is secreted out into the system here, we're going to see this hydrochloric acid that's actually going to aid in cleaving pepsinogen into pepsin, and now we can start breaking our proteins down. Okay, breaking those peptide bonds between the amino acids and the proteins. So let's look at the production and secretion of this hydrochloric acid. Okay. We got the cells here, the parietal cells. Right, these are the ones that are going to make the hydrochloric acid. You can see proton or potassium ATP pump moves protons in to the lumen. Okay, how does this get created? We'll see we have carbonic anhydrase, which we saw in respiratory system. Carbonic anhydrase is going to take CO2 that is in the cell with H2O and make carbonic acid. As we make carbonic acid is going to ionize into our protons and bicarbonate. And almost like what we saw in our red blood cells, we have a shift of chloride in. We have a exchange of bicarbonate with chloride into the cell. Okay, so we're going to have hydrochloric acid. That is what's going to be secreted out into the lumen here. Okay, but it does get converted from CO2, and what you can see is as we make this acid, we are putting bicarbonate into the bloodstream. So if we lost a lot of acid, if we were throwing up and losing acid, we have to replenish this acid. We're going to increase this activity, and what are we going to be doing? We're going to be putting more bicarbonate into the blood. So we actually, when you are vomiting, you have to replenish this hydrochloric acid. You're actually going to make the blood a bit alkaline, because one of the byproducts is that bicarbonate gets brought into the blood for each hydrochloric acid molecule that we are secreting into the lumen. So looking at what are the roles, why do we have this acidic environment? Okay, it can be hostile on the cells that line it, but so it must serve a function, and several of those functions are: the low pH is going to denature the proteins. Remember, these proteins are well folded up. We have to get at the bonds between the amino acids to start breaking down those proteins. So one of the things is we have to denature the protein. We have to have it out on that chain, so those enzymes have access to those bonds to be able to cleave them. Okay, remember, the enzymes have to bind the substrate to be able to convert it. Also, the acidic condition helps in forming pepsinogen. Remember, pepsinogen is inactive. Okay, the HCl is going to make the pepsin active and help cleave pepsinogen into pepsin. Right? Once pepsin's active, then we can start chemically breaking down those proteins into shorter peptides, smaller and smaller chains. Okay? And as we said, the pepsin, one of the functions of the pH is to increase the activity of pepsin here. Okay, so that basically that low pH is going to aid in our chemical digestions of our proteins here. So once we move through the stomach, we're going to move, after moving through the stomach, we're going to move into the small intestines. Small intestines is that windy bunch of tubing, predominant in the abdominal region. All right, and if you unfold it and laid it out, it'll lay out about 20 feet long in length. Okay, so we have this numerous folding. It's separated into three stretches. We got the duodenum, the jejunum, and the ileum. Right. And this is where we're going to get further breakdown, mainly chemical breakdown, chemical digestion of the foods. And then we're going to get the bulk of the absorption is going to take place chiefly in the jejunum. All right. What we're going to see, we're going to talk a bit about this because many of the accessory organs are attached to the duodenum, because we're going to get secretions from the liver, the gallbladder, and the pancreas here. All right, so we'll talk about this in a bit, looking at these secretions, but much of what is secreted in the duodenum are enzymes that are going to be involved in that chemical digestion. Okay. So as we move through the stomach into the small intestines, we move through, and this is where we're going to actually get the absorption to take place. Okay, we're going to break those materials down, and we're going to get the absorption. So we're going to look at functions of the duodenum, or the functions of the chemicals that are released in the duodenum from either the liver, gallbladder, or pancreas. And then we'll look at the actual absorption, the process of absorption taking place here. So let's look at the anatomy of the small intestines, because the form will give us the function, but this should look familiar from your anatomy days in your lab. You saw the model looking here, and basically when you look at the small intestines, you can see there are these circular folds. Okay, the lining is folded up, and then in these villi here, these folds Okay, there's villi. And then on top of those villi are these microvilli. Okay, these smaller little, and it's called what makes up the brush border. If you get a high enough microscope, it looks like this little brush-like movement moving through there. Okay, so we have a circular fold with the villi on it, and then on the villi we have these microvilli. So what this is giving us is it's giving us a very large surface area. This large surface area allows us to diffuse things in more readily. And what are we doing? We're diffusing or absorbing food. We have to bring food in, diffuse it in. Okay? And this surface area is going to allow us to be able to successfully absorb. So when you see this is the small intestine, and it is set up this way so we can get sufficient absorption. As you can see, there is a large blood supply here, and also there is lymphatic vessels going up. Okay, this is where we're going to absorb the food or the nutrients into. As I said, those microvilli that are on top of the villi that are on top of the circular folds, there are enzymes embedded in the membrane here. Okay, these brush border enzymes. These brush border enzymes are going to be involved in basically finishing up the chemical digestion. Or the chemical digestion is cleaving the bonds between the individual molecules. For starches, it would be saccharides. Okay, carbohydrates, individual carbohydrate molecules. Okay, for proteins, it would be breaking bonds in those peptide bonds. And then we'll see for lipase, it would be breaking down the bonds in lipids. But these brush border enzymes, okay, you can see have some activities. They're going to break down disaccharides. Okay, disaccharides being a one carbohydrate, two carbohydrates. Okay, so the function of these brush border enzymes for here is to break this bond to get that carbohydrate and that disaccharide into two monosaccharides. Right? They are not bonded anymore. So that is the function of the brush border enzymes for here. Peptides, they're going to take peptides. Peptides would be amino acids, chains of amino acids. They're going to break these down even further. Okay, so basically the brush border enzymes here are here to finish off the chemical digestion. And now, once we get into these smaller components, we can get proper absorption. Okay. One common brush border enzyme or common disorder, people that are lactose intolerant, where these enzymes are proteins. So there has to be a gene for them to create them. If there is a mutation in the gene, you may have the lack of lactase. Lactase takes lactose, which is a disaccharide, and breaks it into individual components. All right. If you don't have lactase, you cannot break this bond, and it stays this way. And unfortunately, you cannot break this down. And this cannot be absorbed. You have to be, for carbohydrates, you have to be in a monomer, in an individual molecule to be able to properly absorb. So therefore, people that lack lactase aren't able to break down, and they don't absorb the lactose, and it causes disruption in the GI tract. So after we've done our bulk of our absorption, we're going to move ourselves in. We're going to move that food into the larger intestines, the large intestines, right? We've made our way through here, through the duodenum, through the jejunum into the ileum, and then finally we're going to make our way. We're here at the start of the large intestines. Okay, we're going to go back. We're going to talk about the processes, because remember the duodenum, we're going to be talking about the liver and the pancreas. We'll talk about the secretions here that help aid in digestion, and we will talk about the absorption taking place in the small intestines. But we're going to continue our jaunt through, so we'll walk our way through the large intestines, and then we'll jump back to these processes taking place here. Okay. So we've absorbed most of the large macronutrients through the small intestines. In the large intestines, we're going to absorb some of the fluids and ions. All right, and we have material that is left over. We have some waste materials. As we move our way through here to the large intestines, all right, we're going to make the feces with the waste material. We're going to store it, and we're going to bring it to the rectum. Okay, the rectum is the last stop before we go through the anus and out the body and hopefully to the toilet. Okay. Along the stretch here, there lies many bacteria live in the large intestines. Okay. These are good bacteria. Okay, this is the probiotics you hear about that people take. People drink yogurt because it has good bacteria in it, and these bacteria serve functions in helping to produce certain vitamins, facilitate the water absorption, and they are also the ones that are basically going to produce most of the gas you make, because they're going to break down those plant materials that we don't have enzymes to break down, but the bacteria do. And basically, as they do it, they create gas. So many of the fibers we can't break down, but some of the bacteria have the enzyme to break down some of those fiber materials. And when they do, they create the gas. Okay. So we've made it through the large intestines. We've got into the rectum. That's the last stop. And then basically, after we reabsorb, after the pressure, there is enough waste material, feces built up. Right, that's going to result in defecation and basically going to the bathroom. As I said, we're going to jump back and look at some of the functions of those accessory organs. One of the larger of the accessory organs of the digestive system is the liver. One of the chief functions in the digestive system is it's going to aid in lipid digestion. It's going to do this by producing the bile. Right? The liver also involved in detoxification. When you go out to the bar on a drinking night, the liver is going to be one of those that is detoxifying those effects of the alcohol. It is also one of functions in intraconversion of nutrients, moving between different materials, making them into glucose and so forth. And it also involved in storing the nutrients, synthesis of blood, and also phagocytosis. But for our purposes, basically the main function we're looking at for the digestive is it's going to create that bile. That bile is going to help do what we call emulsify the lipids, so we can successfully digest the lipids and be able to successfully absorb them. One of the things you probably learned in anatomy was the hepatic portal system. So the nutrients, we're digesting the nutrients and then absorbing them into the body. The absorption takes place around the stomach. There are capillary beds that line the digestive tract of the small intestines. But what's going to happen is we have a portal system. We have a capillary bed around the stomach and the intestines in which we get absorption. It's going to go to the hepatic portal vein. And the first stop, at least for proteins and for carbohydrates, is the first stop is going, or at least for amino acids and carbohydrates, is going to be the liver. So it goes to the hepatic portal vein, and then into a capillary bed that will feed into the cells of the liver. We'll see the lipids, the fats go a different route, but those of the amino acids and the carbohydrates, they are going to get absorbed at mainly the small intestines and then brought over to the liver for first processing before they make their way out into the general blood supply. So when you cut in to the liver, you'll see the cells are arranged in these lobules. Okay, these hexagon-shaped lobules. And you can see there is a very rich blood supply, and we have several branches. At the edge of these lobules, you will find basically 3 vessels or ducts along the way, called the portal triad. Okay, you can see here at this lobule, this lobule, this lobule, they all have these 3 vessels or ducts. There is the hepatic duct. That is the green, because remember, one of the functions is that the liver is going to make bile. Okay, so radiating into these lobules are rows of the hepatocytes, the liver cells. Okay, and the liver cells are going to make bile, and they're going to secrete it into these little green caniculi that feed into these hepatic ducts. Okay, we have the hepatic portal vein, parts of the portal veins, because this is bringing that nutrient-rich blood that has been around the small intestines, that has picked up the nutrients, absorbed the nutrients. So it is bringing that nutrient-rich blood in this direction to these lobules. And then the hepatic artery is going to supply, that's the red vessel here, going to supply the oxygen-rich blood for these hepatocytes to use. Okay, these all merge together, or at least the blood supplies merge together to form this sinusoid. So in this case, the blood is coming in this direction and then in to the lobule, and in between the lobule, okay, are these central vein. Okay, so the oxygen-rich blood and the nutrient-rich blood from the small intestines are going to merge together, sinusoid, to the central vein. Okay, ultimately central veins unite and bring the material to the inferior vena cava, into the systemic blood supply. Right. And that's those hepatic cords. The hepatic cords are the cells, the hepatocytes, that line here, make these cords. Okay. They have the hepatocytes, and they also have what's called the biocaniculi, that are these little green vessels here, or ducts. Okay. Hepatic sinusoids, as we said, is where the oxygen-rich blood, the red vessel, and the blue vessel, the hepatic portal vein, merge to bring the blood through. Okay. Along this here, along the sinusoid, lines the Kapuffer cells, because remember, we're bringing stuff from the outside environment. It's also going to contain pathogens, and so these are phagocytes to start eating up any foreign bacteria or foreign material, foreign pathogen that we are bringing in when we eat our food. So we saw those hepatocytes. They make that bile, they excrete it in the biocaniculi, all right, and then put it in the hepatic duct. The hepatic ducts all merge together to ultimately merge into these right and left hepatic ducts. So all the cells through here are making bile, and they're going to eventually end up here. Okay. The gallbladder is the storage for that bile that is created from the liver. Okay. Doesn't create it, the liver creates the bile, brings it into those hepatic ducts, and then if not needed, if you're not eating a fatty food, then it is stored in the gallbladder. The bile is going to aid in the digestion of the fats. And we're going to see that process when we look at the absorption. But if you think about it, fats, what do fats? Fats are going through the stomach, okay, going through the lumen, and it is an aqueous environment. The fats do not want to spread out, right? They want to glom together because they are non-polar. Okay, so that is the function of the bile, because we are using enzymes to break the bonds between that glycerol and the fatty acids, and to do that we have to have the fats basically dissolved out so the enzymes have access. Otherwise, if the fats are all glommed together, the enzymes aren't going to have access to those internal fat molecules. Okay, so that is what the emulsification is to reduce the attraction between the fat molecules and allow them to spread out and basically dissolve so the enzymes can come in and break down the fatty acids. When you eat a lot of fat, CCK is a hormone that will be released, and that's going to stimulate the contraction of the gallbladder. So when you eat a fatty food, the gallbladder contracts, right, and it secretes the bile. Many people have had gallbladder surgery to remove the gallbladder. What they definitely recommend you not to do after having a gallbladder removed is eating large fatty foods or fatty meals, because your body has to adapt to be able to create enough bile, right, since you don't have this storage anymore. So you may not get proper digestion of fats if you eat a large fatty meal after getting your gallbladder removed. So here's the general flow, because the biliary apparatus, the apparatus that's going to move the bile, but ultimately that bile is going into the duodenum. Okay? It's going to be secreted into the duodenum. Right? The general flow from your anatomy days is we have the hepatic ducts. These would be coming from all those ducts in the liver that have merged together on the right and the left of the liver, right? And they will come down common hepatic duct. If you're not eating food and you don't need a lot of bile, it can be reverted back up and stored into the gallbladder. When you eat a fatty food, boom, CCK stimulates contraction. Now we push out through the cystic duct into the common bile duct, which will ultimately lead to the duodenum. Okay, and now we get bile mixed with the fats. We get emulsification, and those fats can dissolve out, and now we can get proper digestion of the fats. The other large accessory organ for the digestive system is going to be the pancreas. Pancreas has a very functional role in here, in that it is going to produce most of the enzymes that are used for the chemical digestions. 20 plus enzymes that are used to break down the lipids, break down the amino acids or the peptides, and break down the carbohydrates or the starches, okay, what's left of them. Okay. And you can see here is that bile duct from the liver coming into the duodenum. Again, this is a stretch of the small intestines, that first stretch called the duodenum, right, which ultimately leads into the jejunum. Okay. But the pancreas, 99% of the cells here are serve for exocrine. serve for the secretions that are going into the duodenum here. And the other 1% are part of that endocrine function, and they make that glucagon and that insulin. But for our sake here, we're looking at the pancreas for its exocrine secretions into the duodenum. You can see the cells of the pancreas will be making the various enzymes and also bicarbonate, which we'll see in a sec, and they feed into ultimately into the main pancreatic duct. The main pancreatic duct is going to basically secrete its substances into the duodenum. And again, many of the substances that form up what we call the pancreatic juice is going to be all those various enzymes that are going to be used for the chemical digestion of the food, along with also bicarbonate. Because remember, this first stretch here is receiving, this duodenum is receiving the contents from the stomach, which is a highly acidic substance. Two main cells, ET cells and acinar cells. The acinar cells that line the duct that are going to feed ultimately into the main pancreatic duct make the zymogens, make the different enzymes. All right, at least 22 different types of enzymes being made here. Okay, and again, they are zymogens because they are breaking down lipids, peptides, carbohydrates, which exist in the cells, and you don't want an active enzyme running through the cells, so they will secrete those zymogens into the ducts here, and ultimately those will lead to the main pancreatic duct, and into the duodenum. Okay, the ET cells then are going to produce that bicarbonate fluid. Okay. Bicarbonate is going to be there because the duodenum is receiving the contents from the stomach, the highly acidic contents, and so the bicarbonate is there to neutralize that highly acidic substance coming from the stomach. So that pancreatic juice, as we said, is going to contain the enzymes. It's going to contain the bicarbonate to help buffer that acidic material coming from the stomach. Some of the major enzymes coming out are trypsin. Okay. They're going to digest proteins, but there are many other, or there are about 22 plus enzymes being secreted by the pancreas. So there are many other proteases, enzymes that are going to break down peptide bonds and those peptide chains. Lipase, we saw that in the lab activity. That's going to be involved in lipid digestion. And then amylase. Amylase is going to break down those starches. Okay, we already saw there's amylase in the salivary amylase back from the parotid gland. So we start the actual starch digestion in the mouth, and we continue it when we secrete more amylase in the from the pancreas into the duodenum. So now we'll look at the secretion of bicarbonate, and then we're going to look at the activation of the enzymes from the pancreatic juice, the juice that the pancreas secretes in the duodenum. So the bicarbonate, again, it is being formed to neutralize that acid, the acidic material that is coming from the stomach, because it is the next in line. It's stomach and then duodenum. And two ways in which it's being made or secreted into the pancreas in the pancreatic duct is those cells that line the duct. There is, you see, one way is through co-transport with sodium. Bicarbonate is being brought into the cell and then into the lumen, and also CO2 is being brought into and making carbonic acid with our carbonic anhydrase. Right? And then as we create carbonic acid, it's going to ionize into bicarbonate and into the proton. And the proton gets secreted back into the bloodstream. So you can see these cells, what they're doing is they're pulling out bicarbonate and they're also returning protons. So if these cells, say you have diarrhea would remove the contents through here very quickly, and so these cells would have to replenish this bicarbonate. If they're doing that, they're continually pulling out bicarbonate from the blood and also bringing in protons into the bloodstream. So with extreme diarrhea, you can see you would start making the blood more acidic. Now the other component of that pancreatic juice is the enzymes. Remember, these enzymes are breaking down carbohydrates, breaking down proteins, breaking down lipids, all right, which you would find in the cells. So many of the enzymes that are released by the pancreas are released as zymogens. They are inactive molecules. And so the pancreatic juice gets released, but if you remember on the small intestines, there is those brush border enzymes. Okay, and some of those brush border enzymes will convert trypsinogen, sorry, there should be a P here, trypsinogen, and cleave it. Here's the enzyme cleaving it and making trypsin. Trypsin is a protease. It's going to break down or cleave other proteins. In this case, this trypsin, not cleaving the food molecules, but is cleaving the other zymogens, the other inactive enzymes. So when it cleaves, it can create the active enzymes. So that's how we're going to get this activation of these enzymes, since they are released as the inactive form. Finally, before we move on to looking at the absorption, let's look at a couple of the main hormones that are going to help regulate the digestive process. Two, I want you to know gastrin and CCK, cholecystokinin, right here. Okay, we saw that earlier on a slide, and that it stimulates the gallbladder and secretions of the pancreatic juice. CCK, usually when you eat a high fatty diet or high fatty meal, you go to In-N-Out and eat a 12 stack. You got all that fat in there, you will be definitely secreting CCK. One of the other major hormones for digestion, gastrin. And gastrin is going to regulate the release of that hydrochloric acid and pepsinogen. So it's going to act at the stomach to help release those structures or release those compounds. Alright, so now we've gotten through digestion. We've broken down the food mechanically, we've broken it down chemically, and now we can get proper absorption of these nutrients. We'll first look here at carbohydrates. Remember, starch. Starch is that long chain of glucose, or a monosaccharide. And what we saw is that amylase. That amylase in the saliva, the amylase in the pancreas are going to break down those chains, right, into smaller chains. Then those brush border enzymes are going to break it down even further. And for carbohydrates, we have to get down to the individual molecule. We have to get down to the monosaccharide, which is glucose, galactose, fructose. Okay, we can't have a disaccharide. We can't have two glucose molecules bonded together and get absorption. We have to get these carbohydrates down to the monosaccharides. Okay? And once we do, in the small intestines, that's where we're going to get the bulk of our absorption. Most moves through with sodium. Right? We get co-transport with sodium, secondary transport. Sodium is going to move in, its concentration gradient, moving glucose and galactose in. Okay, some, like fructose, have diffusion through the channels. Okay, but once in the cell, then they get absorbed into the bloodstream. Okay, but the key is we are in this form. Remember, if you don't have that lactase enzyme, that lactase is a disaccharide, right? It won't get absorbed. Lactase cannot be absorbed here. It has to be broken down into its individual monosaccharides. Proteins are similar in absorption as the carbohydrates. The amino acids, you can see they get broken down, those pepsins, all right? There's these exopeptidase. You can see those break down the N chains. The endopeptidase, they can break down anywhere in the middle, and then you also have those brush border enzymes to break down those smaller chains even further. Most of the absorption of amino acids is similar to carbohydrates in that it's going to be with co-transport with sodium. The difference between the amino acids and carbohydrates is some amino acids can be absorbed in di form or even tri form. But once those dipeptides or tripeptides are brought into the cell, they are further broken down into the individual amino acids. So getting absorbed into the bloodstream is the individual amino acids. Okay, but unlike the carbohydrates, we don't have to be completely broken down to individual, but once in the cell, they will be broken down into the individual amino acids here, all right, to get actual absorption into the bloodstream. So you can see we got to get this proper digestion. We have to have all these enzymes and the proper digestions to get the proper absorption of the nutrients. Finally, we got the lipid digestion. Okay. And you remember we have that bile that's going to help emulsify the lipids. Okay. And if you remember our lipids, the lipids are formed. We got the triglycerides, we have the glycerol backbone, and then the long carbon chains, the fatty acids. Okay. And the lipase. Lipase enzyme is going to work and break down the triglyceride into a monoglyceride, so a glycerol with one fatty acid attached, and it's going to cleave off two of the fatty acids. Okay. As that happens, these components here are going to be packaged into a micelle. Okay, a little ball forming with these non-polar molecules. The micelles is what actually gets absorbed into the cells of the small intestines. Okay. When it makes its way back into the cell, those monoglycerides are converted back into our triglycerides. Also in the cell, those triglycerides are going to aggregate with cholesterol and proteins and other phospholipids, and they're going to form what we call these chylomicrons. Okay, from here, chylomicrons, and then the chylomicrons are what are actually going to be absorbed into the body. But different from the amino acids and the carbohydrates, amino acids and carbohydrates will be absorbed into the bloodstream here and then brought over by the hepatic portal vein over to the liver, whereas the chylomicrons are going to move into the capillaries but then ultimately move into the lymph. So they are absorbed into the lymph. So they don't go straight to the liver for processing. They make it from the lymph into the bloodstream, and then they can be brought to where they need to go nutrient-wise in the body. So we can see once we get the fatty acids into the bloodstream, if you remember, fatty acids or triglycerides are nonpolar, so they don't want to be dissolved in the blood, so they are transported with these chylomicrons. Basically, this chylomicron has phospholipids surrounding it and has these lipoproteins. Okay, and so this chylomicron gets absorbed, it's in the bloodstream, and now lipids can be transported. There are what we call these lipoprotein lipases that bound onto those apoproteins, and then the triglycerides can be removed and brought into the cells. Another form of this, that lipoproteins are actually not absorbed through the bloodstream but transported through the body, and they are made by the liver, some endothelial cells, and we're just going to touch on these because from a medical standpoint they are pretty relevant, and that you guys always hear good cholesterol, bad cholesterol, but your liver will create for transport of cholesterol and fatty acids. We have these very low density proteins. Okay, they're going to transport glycerides and such to the different organs, and one of the ones you hear, LDLs, one of the common blood tests, is that they are the bad cholesterols. They are going to transport cholesterol. They're made in the liver, transported cholesterol to the various organs, okay, the heart, the skeletal muscle, but also the blood vessels. And that's where you get those plaques. Right. This is in the blood vessel. Okay, you can see how this limits the transport of blood. On the opposite end, you hear good cholesterol, HDL. Okay, and it actually goes in reverse direction of the LDL is going to transport from the liver to the cells, okay, or the tissue, where HDL is actually going to do the reverse transport. It is going to pick up these here, and the excess cholesterol, and then transport it back to the liver for processing, where the liver is going to eventually turn it likely into bile. Okay, so LDLs going to transport, they are the ones that deposit those plaques, where HDL would bring up. So you see in our blood tests, it's not always, hey, I have high bad cholesterol, but it also has, you have to take it into account good. If your good is high along with your bad, that's not as bad a scenario. If your bad cholesterol is very high and your HDL is very low. One of the main ways to get this up is exercise. So among other things, exercise is important for heart and so forth, but it also is good in helping regulate your lipoprotein content, giving you more of the HDL. And so that is it for